All right, welcome everyone. This is the International Society for Vaccines monthly webinar. We appreciate you attending. We have the honor today of having Dr. Gunnveig Grødeland, trying to say my best Norwegian accent, a professor of immunology at the University of Oslo. Her research is located at both UOI as well as Oslo University Hospital. And she focuses on vaccines against viruses such as influenza and SARS-CoV-2. She's been a leader in the Center for Pandemic and One Health Research at the University of Oslo. The center is aimed at performing research enabling improved management of the next pandemic and emergence of new respiratory and emerging diseases. She originally was a Fulbright Scholar at the Scripps Research Institute in San Diego, where she worked on developing vaccines against viral pathogens. And so we're pleased to have her here today. She is a colleague of mine in the Incentive Program, which is a EU and Government of India funded program associated with developing next generation influenza vaccines. So welcome. If you want to share your screen, you can begin. Thank you. Just unmute yourself. Thank you.
Hi, yes. Thank you. I'm very happy to be here today and share some of the work in my group and look forward to a discussion afterwards. So I will today focus on vaccines for managing the next pandemic. And I think as a starting point, I would like to remind us all about the WHO recommendations of 2018 on the topic of which priority diseases we should focus current vaccine development on. Now, for pandemic preparedness, we have been waiting for a bird flu now for quite some time. But you can see here the diseases on this list were also diseases expected to be quite potentially problematic for the future. Now, you can see at the bottom of this slide that besides the Crimean Congo hemorrhagic fever, Ebola, Lassa, MERS, SARS, Nipah, and Nipah viral, Rift Valley fever, Zika, and disease X, you can see that there were some other diseases and viruses that were discussed as relevant by the WHO, but considered less likely to be the next pandemic threat. One of these diseases on this list is, as I have highlighted here in red, the highly pathogenic coronaviral disease other than MERS and SARS. And this is what we got. I'm using this starting point to demonstrate the difficulty in trying to predict which virus will actually cause the next pandemic. And even if we could so, which variant of that virus is likely to cause the next pandemic is even more difficult to predict. We have now coming up as a likely candidate for the next pandemic emergence, as you know, H5N1 influenza. But I think this is a reminder that it might be something else entirely that actually is the next thing we have to prepare for. So for prevention of the next pandemic, I think our challenge as a starting point is that it is difficult to predict which virus variant will emerge. So the only thing that we know is that vaccine stockpiling in advance is probably a pretty poor alternative. We have two potential solutions for that. We can either base our strategy on rapid development of variant-specific vaccines that can induce neutralizing antibodies to prevent infection and disease with whatever virus comes next. Or we can try to develop vaccines able to broadly protect against many different viral variants of those viruses that be considered more likely for the next pandemic. I here would like to take just a minute to think about how we are going to focus these vaccines. Because current vaccine development is focused a lot on development of neutralizing antibodies against a particular virus. And while this is the best type of immune responses for preventing infection and mild disease, I would also like to remind you that we have a lot of other effector mechanisms for antibodies. They may contribute to opsonization and phagocytosis of microbes. They can raise ADCC responses or contribute to complement activation or phagocytosis in association thereof. A common denominator for all of these other mechanisms as compared to neutralizing antibodies is that they are less sensitive potentially for antigenic drift in the virus that we are dealing with. That means neutralizing antibodies are the most sensitive to changes in surface proteins, the most likely type of immune responses to first lose their efficacy, but that does not mean we're left with nothing. Besides all of these other antibodies, these other antibody effector mechanisms, we of course also have T-cell responses that are less prone to lose their efficacy upon antigenic drift. If we go back to the current scenario, neutralizing antibodies represents the strategy that most vaccines have today with respect to kind of trying to quench the pandemic emergence that comes next. In our attempt to rapidly quench a new virus emergence, we have already a lot of research that have been going on the past decade into different subunit vaccines, for example, DNA and mRNA, as depicted here. You can see on your left-hand side the results from a clinical phase 1 trial with a spike from the coronavirus MERS being tested and published in 2019. And you have on my right another example with a clinical phase 1 trial with an mRNA vaccine this time and developed against influenza H10. These are just some examples to kind of illustrate the massive amount of research that has gone into subunit vaccines for the past decade. And then, as you know, we had the pandemic emerging around 2020 and henceforth. We then really demonstrated the power of that research that had been done previously. We had rapid development and approval of particularly mRNA vaccines against SARS-CoV-2. And the pandemic really demonstrated that these new formats can be rapidly produced and mass deployed for protection of the global population. We can discuss the speed and we can discuss how it was deployed, but you know, it kind of worked as a starting point. I would then like to focus on some of the research that's going on now in my group that has been started many years ago, because novel vaccine development has a tendency of taking some time. So we have been focusing on DNA vaccines, aiming for rapid quenching of a new pandemic threat. The challenge that we decided to tackle was the reduced immunogenicity often associated with subunit vaccines. We decided to focus on influenza. So here you can see that we took the surface protein of influenza, hemagglutinin, and inserted this into this vaccine structure, where we linked genetically the influenza hemagglutinin to a dimerization unit, which again was then linked to a targeting moiety that could either be single chain FV against the MHC class 2 molecules or natural ligands. I said we delivered the vaccine as DNA, and then of course the cells at the site of vaccination would take up the DNA and produce the corresponding protein. And that protein with this yellow targeting moiety would be steered specifically to antigen-presenting cells, leading to uptake and presentation of peptide on MHC class 2 molecules, where the peptide could be recognized by T helper cells. We have the hemagglutinin in the full-length conformation, which means that this can also, on the protein, be recognized by B-cell receptors, and we can have rapid development of antibodies. I think also an important feature is that targeting towards APC with this vaccine protein will lead to a cross-presentation and activation of cytotoxic T cells. I think a pretty cool thing, still, is that depending on the target you select on the antigen-presenting cell, you can steer responses to different types. That means if you are targeting your vaccine towards MHC class 2 molecules, for example, we know that you will get a dominant antibody type of response. If you're targeting chemokine receptors, on the other hand, like XCR1 or CCR1, 3, and 5, for example, then we know that the response is more polarized towards cellular immunity. It's not black and white. You will get a little bit of both with both strategies, but it's kind of the dominance that you are steering towards different types here. So with this vaccine and for influenza, we decided to focus on steering of the vaccine antigen towards MHC class 2 molecules. The purpose being that we wanted to raise as much antibodies as possible. Neutralizing antibodies would be favorable, with the aim of actually trying to quench viral spread in a potential population. After some nice results in mice, we progressed as depicted here to vaccination of ferrets and pigs. And you can see the HI titers after vaccination in these animals. The black bar corresponds to vaccination with this APC-targeted vaccine. And then in the white bar, we have a corresponding vaccine that is identical, except that the targeting moiety, the single chain FV against the MHC class 2, was replaced with a single chain FV specific for the hapten NIP. So identical, but no relevant targeting efficacy for that vaccine. So you can see really from this figure that the selective steering of the vaccine towards antigen-presenting cells is key for getting this high efficacy in ferrets and pigs. We have done some benchmarking of the strategy against the conventional vaccines, and then we chose to compare to pandemics, including the adjuvant AS03. So here, this is in mice. We have to go a bit back to mice for this. In mice, we compared pandemics / AS03, delivered as that you probably know, but it's the vaccine that was developed and used for the swine flu of 2009. So we compared a single dose of that in mice to a single dose with the DNA-delivered MHC class 2 targeted vaccine. Here you can see long-term antibody responses at the top, and you see that there is no significant difference in IgG1 or IgG2A between pandemics and the DNA vaccine. At the bottom, you can also see that about a half year after one vaccination, we challenged these mice with a lethal dose of influenza H1N1. And then you see no difference between the pandemics or the MHC class 2 targeted DNA vaccine with respect to survival or weight loss. So that is good. But you're not really interested in these long-term responses necessarily if you're trying to prevent a pandemic. Then you are more interested in what happens early after vaccination. And so we used the same vaccines, Pandemrix or MHC class 2 targeted DNA vaccine, delivered once. And then here on the left, you see antibody responses on day 7 after this single vaccination. And you can see that there's no difference between these two vaccines. Then the next day, on day 8, we challenged the mice with a lethal dose of influenza again. But here you actually do see significant differences. I see I've forgotten to attach the star signifying a significant difference here. But you can see that the triangles representing the MHC class 2 targeted DNA vaccine actually has improved protection from influenza as compared to Pandemrix with the gray circles. I'm not going to show you what this difference is, but you're going to guess and you're going to be right. It is T-cell responses, indeed, because we do have T-cell induction with the DNA vaccine, but very much less so after Pandemrix vaccination. So our benchmarking in mice also points towards this being a pretty good vaccine for quenching a potential pandemic threat. On that background, we moved forward to ferret studies and then decided to focus on a vaccine against H7N9 influenza. Here you see the titers of neutralizing antibodies on day 26 after a single vaccination. And then we had a booster dose the next day. So on day 40, you also see the enhanced titers of neutralizing antibodies after the second vaccination in ferrets. You can see we used 2 different doses. We used 3 milligrams depicted in blue and in purple 0.3 milligram doses, and then saline, of course. But I kind of like that we got some neutralizing antibodies already after the first vaccination with this high dose group. We challenged these ferrets, of course, with a lethal dose of H7N9. And you can see on the left-hand side that the ferrets receiving saline in green, on day 4, they had approximately a 10% weight loss, indicating the lethality of the dose we gave the ferrets. In blue, you can see that there's no weight loss in the ferrets receiving the high dose. In the low dose, there are some weight loss, less than the saline. And if you look at the clinical signs, you can see that there's nothing in the ferrets receiving the high dose, but there are some in those receiving the low dose and quite a lot more in the saline receiving ferrets. So on this background, and a little bit more that I'm not going to show you, we progressed towards a clinical phase 1 trial with this vaccine. We started before Christmas of 2023 and have now, I think we are about midways in the study. We have 3 different dose groups. We are now in the intermediate dose group. We haven't really started looking at immune responses yet from the vaccinated individuals. But we have so far confirmed that safety is looking good. And the immunogenicity data we are now presently analyzing in my group. So that will be excited, I hope. That is one strategy of how we can quench the next pandemic, trying to make rapidly a new vaccine able to raise neutralizing antibodies against that specific variant. The other one, as I said earlier, was to try to raise more broadly protective responses so that you can avoid the uncertainty of which virus variant will actually emerge. And to do that, you have 3 main strategies. You can focus immune responses to conserve the antigenic sites. 2, You can include more antigens than just the dominant surface protein, such as hemagglutinin, for example. Or you can aim for development of broader antibody responses, as well as T-cell responses. I will now show you some examples from my group for all of these different strategies. The first one being a project where we tried to raise immune responses towards the more conserved part of hemagglutinin, the stalk domain. Unlike most others, we didn't worry a lot about how this would be structurally presented. We just delivered it as a DNA vaccine. In A here at the top, you can see with this DNA delivered HA stalk, we actually were able to form antibody responses that could in ELISA recognize and bind very nicely influenza virus, H1 and H5 as depicted here. You will see that this stem domain, similarly to the pandemic vaccine I just showed, we targeted this instead, the produced protein after vaccination to antigen presenting cell. Here again, using the MHC class 2 targeting moiety, but also using MIP1 alpha, the chemokine, as a targeting moiety. So here we are comparing two different receptors on APC with respect to targeting. In the lower panels, you can see what happened when we gave a lethal dose of influenza H1 to these mice. The saline vaccinated mice lost weight and had to be euthanized for humane reasons. Those receiving the HA stem, either targeted towards MHC class 2 molecules or chemokine receptors on antigen presenting cells, both were protected from disease. That is, when you look at this weight curve, you can see that the first days we do have a decrease in weight for all of these groups. For the APC-targeted vaccine groups, the mice will rapidly regain weight after this initial dip. But I think this illustrates that with the HA stalk, it's difficult to get these neutralizing antibodies and have protection against both infection and mild disease. So I think their strength may more be in protecting against disease progression to more severe stages. Another strategy is to try to, instead of just focus specifically on known conserved epitopes, try to focus more broadly on anything that is conserved between different variants. And so here we are using epitope dilution as one strategy. So you know that there are 18 different types of influenza A, and each one characterized by being so different from the others that antibodies will typically not bind between groups. Truth, of course, with modifications, because there are some conserved epitopes. And so our strategy was to make a mixture of many different variants with the aim of up-concentration of these shared epitopes, and at the same time diluting these regions that were specific for a single hemagglutinin in this case. In the first attempt, we made a mixture with 6 different hemagglutinins from 6 different subtypes, as depicted here. And demonstrated that in this mixture, we could still have strong antibody formation against all of the subtypes included in the mixture. So that's important as a starting point. We also saw that we had, if you see at the top, we had some levels of antibodies against the hemagglutinin from H1N1 influenza. This is a subtype that was not in the vaccine mixture. And we also saw that these antibodies could not neutralize H1N1, but they were certainly there. When we then challenged, this is in mice, so we challenged the mice with a lethal dose of H1N1. At the in E, you can see E and F, you see challenge with the H1N1 Calo7. And in G and H here, you can see challenge with the variant PR8 from 1934. We saw that even though we got only low levels of antibodies against the HA from H1N1 in this mixture, we actually had very good protection against the H1N1. We demonstrated that this protection was partly mediated by T-cell responses and partly by broadly binding antibodies that were non-neutralizing. Based on this study, we progressed with development of a larger mixture to really force even more the development of antibodies against shared epitopes. And so we made a mixture with hemagglutinin from either 17 or 18 different subtypes. Here you can see the different vaccines that we made. So far, you can see that all of the vaccines have this X-shape structure. And on your left, you can see what I'm calling the AB mix. And here, this dimerization unit that we included is making sure that in this X form, you will have two separate hemagglutinins presented on the different arms. So you're securing heterogeneous dimer in this case. On the right-hand side, we decided to continue to use a CH3-based dimerization unit. But in this X structure, you don't have control over which HA will go to which arm. So when we give this as a DNA vaccine in the cells at the site of injection, you will have a random formation of these dimers. Most of them will have two different HAs presented on the X structure, but some of them will also have bivalent display of two identical HAs. And we wanted to test how this difference would also pan out in mice after vaccination. To make it a bit more complex, we also decided to make these vaccines targeted towards different receptors on antigen-presenting cells, either steering towards chemokine receptors or steering towards MHC class II molecules, or as you can see at the bottom on the two sides, steering towards the hapten NIP. So this is the non-targeted negative control. Firstly, importantly, when we vaccinated, we first tested that each of these vaccines independently with the inclusion of only HA from one subtype would raise good antibody responses after vaccination, and where you would have insignificant amounts of antibodies formed against H1 when you vaccinated with the 17 other subtypes. And also we tried to challenge all of these groups with influenza H1N1 and demonstrated that you had no cross protection if you vaccinated with H2 to H18. So that's just background information. Then we made the full mixtures as intended and vaccinated. And here in red, you will see the vaccine mixtures targeted towards chemokine receptor. And in blue, you will see the mixtures targeted towards MHC class II molecules. In A here depicted, you will see that we actually got with this mixture antibody responses against H1N1, even though this HA was excluded from the mixture to really test responses experimentally. You can see in B that the responses were mostly IgG2A, and that we mostly got responses in the mixtures that were steered toward chemokine receptors. We then gave, well, 2 different challenges with influenza H1. In D, you can see that we gave a 5 times LD50 viral challenge. And you can see that there is a significant protection after vaccination with these mixes steering the antigens towards chemokine receptors in red. But there is less protection in the mixture steering antigens towards MHC class II molecules in blue. If you recall from earlier in my presentation, I said that steering towards MHC class II molecules and chemokine receptors in other projects actually has a different type of immune polarization after vaccination. So we get more type of cellular immunity when targeting chemokine receptors, and we get more neutralizing antibodies when steering antigens towards MHC class II molecules. So here we think that that might favor protection in this mixture when targeting chemokine receptors, and that we get a combination of antibody-based protection as well as T-cell-based protection. We have also thought that in a more realistic sense, you wouldn't exclude the HA that you think most likely for protection from your mixture. You would actually include the full range, so all 18. So in this figure here, we are comparing in green a mixture with hemagglutinin from all 18 subtypes to a mixture in red with hemagglutinin from HA2 to 18, but excluding H1. And as you can see, it is indeed favorable to have included the variant or a variant of the challenge strain. So this is a heterologous H5N1 challenge. It is beneficial, but you still have quite good protection even in the absence of a relevant subtype. So if we're going to sum up this study, we can see that epitope dilution can indeed raise antibody responses against many different shared B-cell epitopes, but T-cell responses will also be contributing to protection with this strategy. I think another strategy that I find quite interesting is to use different types of antigens included in the same mixture for raising a kind of broader type of protection by that strategy. And here to that end, we have made a vaccine where we have mixed actually hemagglutinin, neuraminidase, and spike, as well as internal SARS-CoV-2 and influenza antigens, with the aim of making a broad package, kind of multi-layered protection, sort of. So with this strategy, we are seeing that we can get neutralizing antibodies against spike, hemagglutinin, and neuraminidase. And then we also get a layer that awards broader protection because we also induce T cells against these surface proteins. And then as a really safety layer, making sure that the vaccine can award protection against severe disease, we are also raising T cell responses against internal proteins that are highly conserved, as you know, between different subtypes and strains. So this is also one way to think of how we can broadly protect against the variants of the future that we don't know of and that might cause the next pandemic. So I think if we are doing first this summary, I think it is important to be mindful of which purpose a given vaccine will serve. A vaccine aiming for rapid response against a newly emerging virus or a variant should be rapidly produced. And then we know that we can have that both with DNA, mRNA, and protein vaccines. If we are more aiming for broad protection, that opens up, I think, for even more vaccine formats to be used. But these will be important in the sense that you can use the vaccine ahead of a viral emergence to secure basic protection against severe disease. You will then have to lose your aim of potentially protecting against the viral spreading in the population. So focusing really on severe disease, potentially also mild disease, as we saw, with the vaccine formats able to raise neutralizing antibodies. In order to really make the perfect vaccines offering broad immunity, I think we need to know a bit more on the different correlates of protection. It would also be beneficial to know more about antigenic immunodominance. And also, I think one of the things that we should really now try to focus on, particularly since H5N1 seems to be, well, quite relevant for breaking out in the population at some point, we really should look into, in my opinion, how repeated exposures to similar viruses may affect formation of new responses, both in terms of looking at what does our previous exposure to seasonal influenza, what does that have to say for protection against these potentially pandemic variants. I know Tedros has done some nice work there, but also it means how will new antibodies formed, how will they be, what will they be directed against in the context of potential original antigenic sin, as we also know too little about, I think. So there's a lot of research to be done in the future, but I think for the next pandemic, we really need to look at the context and what we want to achieve. Your strategy should be different if it is an emerging pandemic as compared to regular epidemics or a childhood disease. And we should be mindful of whether we are trying to protect from spreading or protect from a disease and against many different viral variants. Also, I think we probably should talk more about how or if our aim should be protection of the individual or protection of society, and in which context these should be aimed differently. So that's how the vaccine is going to be used. But then I also think it's really important to consider who is the vaccine for. And this is because both the risk of disease varies with different groups of the population, as does vaccine efficacy. We know it varies with age, sex, geographical location, in the sense that your immunological evolutionary history, what you have met before of different pathogens or vaccines, will form new responses. We know that the health status will influence responses, and a lot more that we can only presently assume. So to that end, I wanted to bring up some results from Norway. This is from March 2022 and is depicting the incidence rate of hospitalization in Norway caused by COVID disease. And this figure has been broken down into non-vaccinated in gray, people who have received 2 doses of the SARS-CoV-2 vaccine in green, and 3 doses in blue. And then these 3 different groups have been broken down by age. And you can see at the top in the younger age group, well, actually up to 64 years of age, there's very little disease, severe disease, requiring hospitalization in the Norwegian population as caused by SARS-CoV-2. And you don't really see a significant difference between non-vaccinated individuals and people who have gotten 3 doses in this age group. I will have to rush to say that in Norway, we also had some cases of healthy young individuals being submitted to the hospital after a SARS-CoV-2 infection. So I absolutely think that the first 2 doses were needed for adults, also healthy. But these instances were so rare that you cannot see them in the statistics. There is no change to this trend even today in Norway, but I also would like to emphasize, if you look at the age group from 65 to 74, and particularly the age group from 75 plus, we see a very clear influence of each new vaccine dose. So here you see really a clear significant difference between non-vaccinated individuals, people receiving 2 or 3 doses with respect to protection against severe disease. So this, in my mind, really tells us that moving forward, how we argue vaccine usage in different parts of the population, we should be mindful of what it is that we are trying to achieve, not only at the population level, but also at the individual level with respect to protection. Also, as a vaccine researcher, and now also as a vaccine researcher being in clinical trials with my vaccines, I am very mindful, and we should all be, that sometimes, fortunately in rare cases, but vaccines will also in some rare cases induce adverse events. We were reminded of that in Norway when the adenoviral vectors were started being used against SARS-CoV-2, and we actually saw that approximately 1 in 17,000 individuals developed VIT, or this vaccine-induced thrombosis with thrombocytopenia, which is, well, a horrible thing to get. There has also been raised a question of whether the vaccines against SARS-CoV-2 can, similar to the virus, raise long COVID in rare cases. We know that after the vaccination against swine flu of 2009, there were some cases of narcolepsy in genetically disposed individuals. We saw after mRNA vaccination against SARS-CoV-2 that there were cases of myo and pericarditis. Here you can certainly argue that the virus increased the probability of getting that a lot more than the vaccine, but should be kept in mind for repeated doses at least. And I think there are many questions regarding rare autoimmune disease formation after vaccination. An important point is that clinical trials, and we can run them for 10 years, does not matter, they will really not be able to detect rare and potentially severe adverse events. This is something that I think we should argue to the population, that, you know, when you're testing with groups in phase 3 of approximately 10,000 individuals, you cannot detect cases that have a lower incidence rate in the population. And I also think that for many countries, we have been facing vaccine hesitancy in larger or smaller parts of the population. And I think, I don't have the answer, but I think as a starting point, if we are going to meet that, the best way of doing that is with balanced science, explaining why the vaccines are wonderful, how they are intended to work, what their purpose is, but also that in rare instances, they may cause adverse events. And our arguments for why the vaccines are so important to use have to then offer a rationale why it really is important to use them, even though we know that on rare, really rare instances, people might actually get adverse events. Then I think we can also meet the next pandemic potentially successfully. Because in order to do that, we not only need very good vaccines against the next pandemic threat, we also need population trust in the vaccines so that they will actually be willing to take them when we are offering them. So we need to work multidisciplinary, I think, in this respect to reach an improved protection against the next potential pandemic. With that, I will stop and acknowledge that I have a lot of really great collaborators. I would in particular like to emphasize here Dag Kvale, who is my collaborator with respect to performance of the clinical phase 1 trial that I'm presently doing. I have also here emphasized in bold the people who have contributed specifically to the work as presented here. And I thank those particularly, but all of my collaborators specifically. And thank you for listening.
Thank you very much. Enjoyed the presentation, and I particularly like the thought experiments you were putting out there for the challenges associated with developing these type of vaccines. So we have 15 minutes remaining in the hour. If anyone would like to ask a question, all they need to do is raise their hand in the system, and I can unmute you and let you ask your question. So I will start while people figure out how to raise their hand in the system. So most of the studies that you showed today were all in animal models that were essentially immunologically naive to the pathogen or the vaccine you were using. But most humans have, for example, with influenza background, at least with seasonal influenza. How do you think that would affect when people are vaccinated with these subtypes that they've never seen before?
I think that is an excellent question, and one of the things we should really do more research on, because I think that you should have some pre-existing immunity. Even if we have been exposed to H1 and H3 and will meet H5, H7, 9, or whatever it might be, there should be enough similarities so that we have some memory responses formed already, and that might be reactivated by the new subtype that we might meet. But what percentage of the population has that? And what will these antibodies actually be? How protective will they be? What will they target? I think all of that are topics that we know too little about, but I think it's really key to figure it out, because the next time maybe we need to think that, okay, we can accept some disease in the population if people are protected mostly against severe disease, but who do we really have to protect first with vaccines giving neutralizing antibodies? And in order to make those decisions, we really need to know a lot more about the background, what you have as a starting point against all viruses that have some variant circulating in the population.
Okay. Dr. Palese, would you like to ask a question?
Yes, a very, very interesting presentation. I wonder, and you may have said it, how much DNA has to be given. There are 2 hemagglutinin molecules and then the dimerization. So what is actually in the vaccine? Is it one DNA chain? Are there more DNAs? And finally, how does it compare in price? In other words, is this very expensive to produce? Could you please expand on that a little bit?
Yes, indeed. I think that is a great question, because first, if we look at this vaccine structure, this here, this is delivered as one sequence, one plasmid. And where you have encoded the full vaccine in the same plasmid. So in terms of the doses in mice, as a standard, we are using 25 micrograms, not because we can't go lower, but because this is high enough to secure that we get very little variation within groups. testing how low I can go in mice. I know that I can go down to 1.5 micrograms and still get the similar responses in mice, but then my variation within a group increases. So for moving into larger animals, the range set that I have tested in pigs, I have not shown that here, but in pigs I have tested from 25 microgram of DNA and up to 3 milligrams. When I did these experiments, the 25 microgram of DNA was, I thought, my negative control, but instead I actually got quite good responses. After 2 vaccinations, I got good titers of neutralizing antibodies, and after the first single vaccination with 25 micrograms, I actually got quite good antibody levels in ELISA. And that was in pigs that were 50 kilos, Norwegian farm pigs. And in ferrets, you saw that we tested 300 micrograms versus 3 milligrams. And we got a lot better responses with the 3 milligrams as compared to the 300. So when moving into the clinical trial that we are presently doing, I was really, I don't know where to go. And this is why we are doing 3 doses. Our lower dose, we went for a 100 microgram and 600, and then as a high dose 300. I'm very much hoping that we will see good efficacy in terms of immunogenicity, to be more precise, at the doses of 100 or 600 microgram, because one of the arguments I'm usually presenting for this vaccine is also a dose-sparing efficacy enabled by the targeting moiety. So to answer your other part of your question, this vaccine should be cheap and easy to produce. It's 1 plasmid, which is a lot easier as compared to many of the other vaccines today. And it's 1 plasmid in saline, so no fancy thing to formulate it in. As for your question with respect to this mixture, that's a bit different. Because here we have 18 different plasmids included. So for progression of this project to a clinical trial, we would indeed have to do GMP manufacture of 18 different plasmids, which could be a hurdle, yes. So let's see. It has to be really good in order to argue that hurdle. And we are planning now on experiments here in non-human primates. And if they are good enough, you know, maybe it's worth it.
All right, thank you. Dr. Abubakar, would you like to ask your question?
Yes, Ted. If I'm audible, Ted, good afternoon.
Yes, you are. You are.
Yes, I am speaking from Moderator University here in Nigeria. So Professor Grødeland, congratulations for a very good presentation. I have got a couple of questions. If Ted would just give me 1 minute to summarize my questions. Firstly, we have seen in Nigeria that the major problem why influenza is it's still a challenge is the fact that we have different reassorting variants of the virus. Now, Professor Grødeland talked about the combination of SARS-CoV and influenza A virus in one mix. Could that be a form of a syndromic vaccine? Say, for example, a vaccine targeting respiratory pathologies. Could that be? That's on one hand. Then in your clinical trial, Professor Grødeland, I'm wondering, I have seen studies that talked about the influence of the human leukocyte antigens in form of the alleles in response to certain medicaments or vaccines. Are you considering the role of races or tribes within Norway or Europe, where your clinical trial is currently undergoing, to possibly impact the outcome of your vaccine trial? Then thirdly and lastly, this vaccine we're looking at here, or this couple of vaccines we're looking at here, are they serving a purpose of prophylaxis or therapeutic? Because my major concern is the composition of the vaccine being majorly DNA-based vaccines. And we all know the role that storage, especially for cold chain, could do in terms of maintaining the immunogenicity or potency of the vaccine. And Professor Grødeland, I'm going to send you an email privately at the end of the conversation. Thank you, Ted. I'm over.
You're welcome. Gonna, you have about 5 minutes remaining, so see if you what you could answer in that.
I will start answering. Firstly, for this project, as depicted here on the screen, where we are including all of these different antigens, I forgot to say that, but this is a project where we are using proteins, so vaccines in the form of protein. And this is because the population may be hesitant to take DNA vaccines if you're going to be vaccinated every single year. And the intention for this vaccine really is to, similarly to current vaccines, get in updated spike, hemagglutinin, other respiratory viruses, if relevant, to get neutralizing antibodies against these. And then have kind of this protective blanket underneath with T-cell based responses against internal antigens, and they will remain constant from year to year. So that is the purpose here. And I think based on our combined finds from this project, as well as the project where we combined 18 different hemagglutinins, I don't think there is a limit as to how many antigens we really could introduce in this mixture. I think of this in my head as a seasonal vaccine against respiratory disease. We started indeed with coronaviruses and influenza viruses. Could be RSV and other things as well, but you know, that is something we will have to test later. Then your second question was on the clinical trial. Let me think, I got a bit lost when talking. It was on the, would you give me a keyword? Sorry. I have to think.
Yes, if I'm audible.
Yeah. Oh, yes, your HLA type. Now I remember. You asked about the HLA of the vaccine because we're steering the vaccine towards HLA class 2 molecules. And you are right, they are really polymorphic in the human population. But the targeting moiety that we have included on that vaccine, it's specific for a region that's on HLA class 2 molecules in the whole global population. So this vaccine should have the similar efficacy all over the world. When we are vaccinating in this clinical trial in Norway, we are not sequencing HLA as a starting point. So we are thinking that this vaccine should non-discriminately work in the full population. Good. Then you had a third question, and I'm very sorry. I forgot that as well. So a keyword on that.
The possibility of cold chain.
Possibility of cold chain. Thank you. So for DNA, then well, you know, I forgot a sample some years ago in my lab at room temperature. And about a year later, I found it. I have no idea what it had been exposed to. My lab, summer, winter, a lot of different changes. And I thought, you know what? I'm going to test how stable DNA actually is. And I'm just going to inject this in mice and see. And it worked. I saw no difference between vaccination with this plasmid that I had completely forgot about and a plasmid that I knew had been in my fridge and freezer. So DNA is supposed to be really stable, and all of my experience with DNA can confirm that. So I think DNA, as opposed to mRNA, really have solved a cold chain challenge and should be deployable globally, regardless of without the need for anything.
Well, I appreciate you giving this presentation today. Our time is up, and thank you very much. And if this video is actually posted on the ISV website for those who would like to look at it later, or tell your friends and they can watch. Next month, we will meet again. And so until then, good luck. Thank you. Thank you for presenting. Thank you.
Thank you. It was a pleasure being here.